This sample preparation technique is designed to combine the best quality of ultrastructural preservation with the most suitable contrast for the imaging modality in a focused ion beam scanning electron microscope. This protocol is especially useful for samples that require preparation by high-pressure freezing for a reliable ultrastructural preservation, but do not gain enough contrast during the freeze substitution for volume imaging.
For minimal resin embedding, it is essential to remove as much resin as possible to allow proper targeting later on in the focused ion beam scanning electron microscope.
To begin, use the pipette to drip 1 drop of 20% PVP PBS on the cutting mat. Immerse the dissected nervus tibialis in the droplet. Use a scalpel to cut 2 mm length of the sample. Use fine forceps to place the sample in a 0.2 mm deep Type A metal sample carrier. Transfer the carrier into the middle plate of the cartridge. Place the hexadecane-coated Type B carrier on the top as a lid. Close the assembly by lowering the upper half of the cartridge. Close the 3-piece cartridge in the loading station of the high-pressure freezer. Press the process button and proceed according to the manufacturer's operating instructions. In a bath of liquid nitrogen, place sample carriers into cryovials containing 2 ml of frozen 0.1% tannic acid and acetone and close the caps. Place the cryovials into the freeze substitution unit set at -90 degrees Celsius. Start the program and keep the samples there for 100 hours. In the freeze